Hello everyone, I am back again with a new video with the topic the cell the unit of life. Previously we have discussed about the topics like who discovered the cell, when was cell discovered. Let us just recapitulate everything. Robert Hooke was the first to discover the cell. Okay, he observed a dead plant cell of the Spanish oak tree. He took the cork and cut it into thin slices and saw under it the microscope. He observed beehive-like structure and named it cella. From that only cell word was derived. Then Anton von Leeuwenhoek was the first scientist to see a living cell. He basically observed the living cell under the microscope and in the soil or basically rainwater. Okay, he saw the microbe and named it as animalcule. Now next thing is cell theory. Cell theory was basically given by three scientists. Okay, first was Matthias Sheldon, second scientist was Theodor Schwann, and third was Rudolf Virchow. Matthias Sheldon and Schwann basically given the first point in the cell theory, which is all living organisms are made up of cells and its products. And the second point was given by the Rudolf Virchow in 1855, which was all cells arise from the pre-existing cells. Okay, so who was the scientist to complete the cell theory? It was Rudolf Virchow. Who modified the cell theory? Rudolf Virchow. Okay, now we discussed about the prokaryotic and eukaryotic cells also. In the last class we discussed about the differences between prokaryotic and between eukaryotic cells, plant cell and animal cell, and we also discussed about the bacterial cell also. So let us discuss about more about bacterial cell and know the differences about prokaryotic and eukaryotic cells. Okay, let us start with today's topic. Let us first visualize, see the diagram to get it more clear. This is a simple diagram of a bacterial cell. Okay, now let us know about the functions and functions of different parts. First is glycocalyx. Glycocalyx is basically of two types. Okay, solid glycocalyx, which is capsule, and loose glycocalyx, which is slime layer. Let us know about both of these functions. Slime layer basically prevents bacterial cell from drying out, and also it helps bacterial cell to stick to something. Okay, you must have seen some green colored layer on the water, which is basically algae. Whenever you step on it or basically on the rocks, you slip. Okay, so it is because of the presence of slime layer. That slime layer is only present in the bacterial cell. Now let us know about the capsule function. Capsule is a hard covering which helps bacteria to which protects bacteria from the WBCs, and also it helps in colony formation. Okay, next comes the cell wall. Cell wall is made up of peptidoglycan. Okay, after cell wall there is periplasmic space. Periplasmic space is the space between the cell wall and cell membrane. It functions just like the lysosome of eukaryotic cell. Periplasmic space is full of different types of enzymes which helps in digestion. Now, membrane bound organelles are absent in the bacterial cell, or basically we can say absent in the prokaryotic cell, and hence compartmentalization is also absent. Compartmentalization is basically there are different compartments made for different cell organelles present. Okay, not for all. Different chambers are basically there which perform different functions. Okay, now comes next is mesosomes. Mesosomes present in prokaryotic cell is formed by the invagination of cell membrane. Okay, mesosomes can be of three types: lamella, tubular and vesicular. This shape is lamella. Okay, these two shapes are tubular and vesicular. As we were discussing about the functions of mesosome, let us know about it. Main function of mesosome is of respiration. Okay, it helps the bacterial cell in respiration. It just acts like a mitochondria which is present in eukaryotic cells. Mesosome also helps in DNA replication, protein and enzyme synthesis, cell wall material synthesis, DNA copying also, and also in cell division. Now there are two types of pili present in the bacterial cell, longer pili and shorter pili. Just see here, this is the longer pili and this one is the shorter pili. Longer pili helps bacterial cells to get attached to other bacterial cells, or basically helps in conjugation. Shorter pili helps in attachment. Okay, shorter pili is made up of pilin protein. This outgrowth you see is known as flagella. Flagella in bacterial cells is made up of flagellin protein. It is a contractile protein and helps in movement. Okay. Now let us see the table of differences between prokaryotic and eukaryotic cells. First character is nucleus. Nuclear membrane is absent in prokaryotic cell, but it is present in eukaryotic cells. It is because of the histone protein is absent in prokaryotic cells. Histone protein basically helps to compress DNA into small. DNA is very long, and if we open the DNA into a single line, then it could stretch up to 2.2 meters. Histone protein is present in eukaryotic cells, so it helps to compress the DNA in eukaryotic cells. Second character is chromosome. In prokaryotic cells, you will generally see double-stranded circular DNA, which is GC rich. But in eukaryotic cells, you will see double-stranded linear DNA, which is AT rich. Basically, the difference is between the circular and linear DNA. Okay, this is basically circular DNA, and in human DNA, you will generally see it is a straight line kind of DNA. This is basically linear DNA. Now, third difference comes is respiratory enzymes. Mesosome membrane and cytoplasm. Basically, mesosome helps in the respiration in prokaryotic cells, and mitochondrial membrane and cytoplasm in eukaryotic cells. Mitochondria helps in respiration in eukaryotic cells. Fourth difference comes is flagella. Flagella is made up of flagellin protein in prokaryotic cells and made up of tubulin protein in eukaryotic cells. And 9+2 arrangement is absent in prokaryotic cells, and 9+2 arrangement is present in eukaryotic cells. I will let you know in detail about the 9+2 arrangement which is present in eukaryotic cells. Next difference comes is compartmentalization. Compartmentalization is basically absent in prokaryotic cells and present in eukaryotic cells. This is because of membrane bound organelles are absent in prokaryotes and present in eukaryotes. Okay. Next comes cyclosis, which is absent in prokaryotes and present in eukaryotes. Cyclosis is basically circular movement of cell membrane for mixing different types of particles inside the cell. Okay. Seventh difference comes is ploidy. Ploidy is basically the number of set of chromosomes. So now bacterial prokaryotic cells can maximum have one set of chromosomes. Okay, only one chromosome is present, so it is a haploid cell, and eukaryotic cells can have haploid, diploid or polyploid, because they can have one set or even more than one set of chromosomes. Eighth difference comes is vacuole. Sap vacuole is absent in prokaryotes and present in eukaryotes. Prokaryotes have generally gas vacuoles. Okay. Now, examples are cyanobacteria and mycoplasma in prokaryotic cells, and fungi, protista, animals, etc. in eukaryotic cells. That's all for today's cell. Thank you.